and thank you all for coming today. Today it's my privilege to introduce our speaker, Dr. Tracy Heath. She's faculty at Iowa State in Amos. I imagine many of you in this room are probably already familiar with Dr. Heath's work in one way or another. She's one of the world's top computational phylogeneticists today, and as such she looms large not only in the small community of methods developers for systematic biology, but across the greater context of biology in fields that generate or make use of phylogenies. I mentioned she's a professor at Iowa State. There she runs a thriving and growing lab focused on phylogenetic methods development. Before that, she originally earned her PhD under Dr. David Hillis at the University of Texas Austin. She's one of the many luminaries to come out of that systematics intellectual epicenter of the planet today. She's also a postdoc under John Holzenbeck at Berkeley and Mark Holder in Kansas. And throughout this time, from even before her PhD, if you look at her publications all the way through now, she's always been at the forefront of phylogenetic methods development, pushing the envelope, not just in the methods, but even the types of questions we can ask. But apart from her groundbreaking research, she's also very, 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 very active in sharing that knowledge. She teaches in a range of workshops across the world, and some of these are legendary, like the molecular evolution, the workshop on molecular evolution in Woods Hole, Bodega Bay, Applied Phylogenetics workshop there, in the Czech Republic's genomic, what's that? Phylogenomics workshop, and I think Crete maybe, and New Zealand. And then in addition to teaching, she travels around the world, continent to continent, city to city, giving research talks at institutions and universities where she shares her knowledge. So we're lucky today to be the recipient of that. Much of her recent work is focused on divergence time estimation in a Bayesian context, and in particular, working in fossil information into the estimation process. We've done this before, but for the first time, she's developed a method that we can do it correctly. So we look forward to hearing it. Go ahead.
Thank you, Jeet, for that incredibly dear introduction. It's almost sweeter than the introduction I was given when I defended my PhD. Thank you also, all of you, for inviting me. It's actually been a really wonderful visit. It's really amazing to come and have really long, in-depth conversations with people, as opposed to sometimes you go on these seminar visits, you have like 20 minutes, 25 minutes to talk to people, but I had just fantastic conversations, so thank you. So when Lacey invited me, one of the things I told her, this is over a year ago, that I was like really excited about this new project that I had starting, and that I was really hoping that by the time I came to give this seminar, I would have data to talk about this project that I'm working on with collaborators on coevolution in figs and fig wasps. And so if you notice the title, it's not actually about that. Because, so as Jeet said, I'm a methods developer, kind of a theoretician of sorts, and I actually don't have a ton of experience working in the realm of data collection. And so I also learned the important lesson that part is really hard. And there are things like that you have no control of, like companies who buy up patents for RADseq and then closing down the organization that you intend to pay to do that for you. So that has actually been really eye-opening, but we do have now some interesting data coming in, except we just haven't analyzed it. And so that's why I kind of fell back on a different project. But so I did want to start off my talk, though, by just giving some highlights about this project and, some of the things that we're really excited about and interested in, because, I think that there's, probably a lot of people here who'd be kind of keen to think about these types of problems as well. So this project that I have is in collaboration with John Nason at Iowa State, as well as Alan Harry at the Smithsonian, and several other people who are working in the fig wasp mutualism. And specifically, we're really focused on the figs and wasps in the strangler fig clade in Central America, specifically in Panama and on Barrow, Colorado Island. So what we're looking at really is understanding what are the mechanisms maintaining this mutualism in these groups, when you have these pollinators as well as these non-pollinating fig wasps that parasitize the mutualism. And so we have all these mechanisms that could break down this whole system, because you have potentially host sharing and wasp sharing and things like that. And so given that we've observed some patterns that indicate that we don't have this strict phylogenetic, co-phylogenetic congruence between pollinators and their host, then what are the sort of evolutionary mechanisms underlying these patterns? And so these are some of the things that we're looking at. So these are the pollinators in the genus Pegoscapus, and then the ficus strangler figs that are in Barrow, Colorado Island. And so this is the non-pollinating fig wasp that oviposits eggs into the fig from outside of the fruit. And so it doesn't actually enter, and it's relying on the pollination mutualism between the fig wasp, the pollinator wasp, and its host in order to reproduce and carry on, because these things actually, since they don't pollinate, they rely on a fig that has been pollinated. So otherwise the fig will not develop into seeds where they can gall and their young can develop and grow and then mate inside there and leave. There's also a lot of other things going on. So it's an incredibly complex and amazing system. And so we're really trying to get at this question of these ecological and evolutionary processes, sort of governing host recognition that might also lead to introgression and this host switching within these in this group. And so this is just some pictures that my PhD student Kevin Quinteros, who's co-advised by me and John Nason, he's in the field in Panama right now collecting. And so one of the things, the specific traits that we're looking at are these volatile organic chemicals that are used and sort of emitted by the fig plant as a host recognition mechanism. And so we're interested in the composition of these different chemicals on the different species of figs. And that's because when we look at the figs, we see that there are some species that share, within this system, some species that share wasp pollinators. So this is a picture that Kevin sent me of wasps leaving a fig fruit, which is just so cool. But we do see a couple instances where we have different species of figs that have the same pollinators. And so this is a mechanism that we think is there, or that can contribute to patterns of genetic signatures of introgression in this group. And so because of this, we're also interested in the traits that might lead to this type of wasp sharing. And so this is the direction that we're going, and we're just now sort of getting sequence data, and that's why I can't really say much beyond like the broader scope of this project. But if anyone has questions afterward or is interested in talking about it, I'm obviously happy to share more information. So moving on to the regularly scheduled program. In my research, I really have taken two major kind of concentrations. One, now with this amazingly opportunity to do this collaboration on coevolution and work on essentially phylogenetic methods for looking at co-diversification in these type of mutualist systems. And the other is based on a lot of the prior work that I did before coming to Iowa State, and that was really to develop more mechanistic models that reflect how fossils are sampled in the fossil record, how we understand the structure of the fossil record, and how that might be integrated when we're bringing together both data from fossils and data from extant taxa via molecular characters and other things like that, in order to better estimate divergence times and phylogenetic relationships, but also macroevolutionary processes that we're fundamentally interested in when we're dealing with groups and organisms and clades that have fossil records and that we're interested in dating. And so this is kind of one of the interesting directions that a lot of more recent phylogenetic methods have been taking is essentially thinking about things in more mechanistic process-based models that are really considered this generating processes of the data that we observe. And so that's kind of how I've been thinking about the fossil record. And so for phylogenetic inference, I would argue that if we are able to estimate branch lengths in proportion to time, that this is ultimately the ideal situation as evolutionary biologists when we're trying to understand the processes of diversification and evolution. And so in our history in systematics, we've done an excellent job of estimating the phylogenetic relationships among taxa in a sort of non-time-based sense, where in these unrooted trees, we're looking at branch lengths where they're in the proportion of units of substitutions per site. And this is really like this compound parameter, but ultimately we'd like to tease out of that parameter the component that is equal and represented by time. And so because we're interested in, as evolutionary biologists, interested in the sense of absolute time and how lineages might diversify in an absolute time scale, then this is a critical component of what we're trying to estimate in phylogenetics. And so this is where it becomes really important to access data other than molecular data to get at these types of inferences. And when we have inferences in a time, like in proportion to time, either relative time or absolute time, we can then pursue the types of questions that most of us are really keen to understand, like how does biogeography play a role in diversification? How does rates of molecular evolution change across our group of organisms? How do traits evolve in concert with other external factors, or how might ecological sort of variables correlate with patterns of diversification? So these are all questions that we can pursue in a phylogenetic context, but we do need this time scale in order to really estimate this. And in particular, things like biogeography and comparative questions, we really need the time units to be in an absolute time scale where we can really get at, did these events, or does this radiation occur at the time that we hypothesize, like this external factor was driving diversification. And so if we have external data, like data from the fossil record, we can incorporate this in a phylogenetic context. And so here is just an example of a tree based on morphological characters of bears and the stratigraphic ranges of fossils within that group. And so the fossil record itself gives us a structured sort of information about the diversification and persistence of lineages through time. And this is fundamentally one of these really important key data components that we need in order to answer these questions. So that kind of really brings us to this one issue where, for those of us who are trained as neontologists and we're primarily working with data from extant taxa, is that accessing the data from the fossil record becomes a very challenging endeavor to do. And when I started working on these types of methods, I was really starting to talk to more paleontologists to understand, why, how do we build these things into models? And one of the key things I realized is that, oh wait, we speak completely different languages, and we also are like pursuing really similar questions, even though we are doing them kind of in separate kind of parallel approaches. And so this kind of, when I was looking through a lot of this stuff, it brought me to this particular quote that was in this book chapter, and it was really just, I'm not going to read it, even though people tell you when you put quotes on things you should read them, but it's too long. But this really gets at this sense that there's been this general divide between our fields, the fields that are interested in diversification and systematics for many years since the modern synthesis, and a lot of this is driven by the excessive importance placed on molecular data. But I think now we're starting to realize some of the questions that we really want to answer are not going to be solved by more molecular data. Fundamentally, we need more fossil data. If we want to estimate times and absolute times in a phylogenetic context, we need to understand the fossil record better and incorporate those data better. And so what has been nice in a lot of this recent work is there's now been this sort of a methods-based synthesis of our two fields. So of paleontology and neontology then can be encompassed within a single statistical framework that will then at least work to synthesize the data coming from these different fields. And so some of these approaches were nicely described, one by Paul Lewis, which is, he's essentially described a statistical model for how discrete morphological characters will change in a phylogenetic framework. And then Frederick Ronquist, and there were also other papers that did similar things, but this is probably the initial, like, sort of most comprehensive approach to doing this type of analysis, where you're using Bayesian approaches, co-estimating the relationships of fossils and extant taxa and divergence times within the single integrative framework. And so these types of sort of ways of thinking, this sort of paradigm of analysis ends up being a nice way to bring together these two fields. So this is essentially what I'm very interested in now, and since in the last several years I've been working with a lot of people who are developing Bayesian statistical methods for a more holistic approach to phylogenetic analysis in such a way where we can fully integrate over our uncertainty in various phylogenetic parameters. So we're interested in accounting for our uncertainty in the tree topology, the divergence times, as well as aspects of our tree model that might contribute to the generation of fossil occurrence times, our morphological data and the parameters associated with those, and as well as the other aspects of that we might be interested in. So here essentially what I'm showing is a sort of modularized graphical model structure of a fully Bayesian phylogenetic analysis. And the symbolism here, each node either represents a random variable. So in the context of a statistical framework, we often think that our data are random variables that are observations that were generated by some evolutionary process. And given that we have those observations, we can then condition on the probability that we have that observation, given that we have some model that generated that. And that's how we would calculate a likelihood in any likelihood framework, and we use that framework also to in a Bayesian context. And so here what we're saying is that we have a set of morphological characters, and we know that there's some process, some substitution process that generates those characters, including variation in rates across that alignment of characters or matrix of characters, variation in rates of character change across the tree. Same with the DNA data, but then also our fossil occurrence times are considered data under any model, and so, or especially this particular model. And so the appearance of fossils in the fossil record, or our ability to sample those, can come from this generating process that that also will give us the diversification process that gives us our tree of extant taxa. And given that we have this tree, we can link all of these components of our model. So this is sort of just this general kind of really modular idea about how we might put together a statistical model. And so for me, what I've focused mostly on is this idea that we have data from the fossil record, and that comes from some generating process that gives us the diversification of lineages that we see in the present and in the past, and that there's some sampling process built into that as well. And so that's what I've been working on, and so this idea really comes from a lot of work on a statistical model called the birth-death process. And so Tanya Stadler in 2010 introduced a concept of this process where you have a birth-death process is essentially a lineage generating process over time. And given that you can sample that process back in time, then you should have a concept of that model that you can account for the fact that you have sampling of that diversification process. So this is what we've called the fossilized birth-death process, and it gives us this two ideas of trees. So one, we have the complete tree. So given that we have a true complete diversification history of a given clade or life on Earth, we might sample that process back in time and also in the present, where that several lineages are unobserved and unsampled. But given that we can account for the various probabilities that would give us a tree that would give us our sampled tree. So here you see all the dots are sampling events. And we might sample this lineage here that does exist in the present, but everything after that is lost to us because we've not observed it. So if we have observed something, it counts as a sampling observation. And so given that we have a tree like this, and we know the generating process of that and the parameters associated with that, so speciation, extinction, and sampling, we can account for observing this tree given our sample, given our data. And so this is what we're basing our fossil occurrence time model on. And so this is something that we actually presented as an approach to calibrating phylogenies, particularly when you only have fossil occurrence data and you have sequence data from extant taxa. And so the common practice in Bayesian phylogenetics really is to construct these very ad hoc and arbitrary sort of densities that account for our uncertainty in the age of a node, even though that node already has some existing prior that's coming from a different process. And so what we were interested in is kind of getting around this sort of statistically incorrect approach, and one that would also allow us to access more of the fossil record. So our node-based calibrations require us to eliminate a lot of the fossil data from our data set, and given that we know that those fossils are part of the same generating process that gave us our extant taxa, then we can include them into a singular cohesive tree model. And so ultimately what we showed in this paper was that this approach will then give us a more statistically coherent way of representing our uncertainty in node ages, but it also allows us to capture more information from the fossil record. And so under this model, under the fossilized birth-death process, what we're essentially saying is that any recovered fossils are essentially observations of a diversification process that gave us our extant taxa. So since this is how we've always been assuming fossils, even when we're using node calibrations, and this is, it's more appropriate to consider them part of this singular diversification model. And so since these are these observations, we can then compute the probability of sampling this process back in time, given that we have accounting for some model of or rates of speciation, rates of extinction, and rates of fossil recovery back in time. And so this relies on having reasonable interpretations of these things, but given that we do, then we can estimate or calculate the probability of any realization under this model. And so in our applications of this approach, we use Bayesian methods in order to estimate and sort of capture the marginal posterior probability of node ages, given that we've sampled some set of extant taxa and some fossils, as well as having molecular and morphological or morphological data. And so this then gives us this better representation of our uncertainty, say in a node age, conditional on observations of certain fossils. So using this paradigm, we're really interested in kind of capturing that. And so that really brings us to one particular issue that is a little bit difficult to, from a classical systematic approach to really appreciate, and that's this idea of sampled ancestors. So you'll notice that if we sample this diversification process back in time, we'll see that we'll sample lineages that also have sampled descendants. And so these are ancestor-descendant pairs of samples in our data set. And so, among paleontologists, this is not a new concept. And so Mike Foote in 1996 published this paper, that essentially illustrated that there is some probability of having sampled ancestors, given that we have some reasonable rate of sampling back in time, sampling the fossil record. And so what he's shown is that there's this non-zero probability of obtaining sampled ancestors in a data set that samples back in time. And so what what I want to just kind of bring home a little bit is why that is an issue and or a non-issue if you account for it. And so, given that we have some idea of what our complete phylogeny is, if we're sampling that phylogeny back in time, so we're saying that fossils are part of the same process that gave us extant taxa, that they belong in a tree together, and if we're sampling that tree back in time, then there's going to be some non-zero probability that we sample a lineage that also has a sampled descendant. And what this really plays into is especially the rate of sampling taxa in the present and the rate of sampling fossils back in time. So here's our complete tree where we have the entire process, and then we have our reconstructed or sampled FBD tree. And so you see there are several cases where we have lineages that we sampled that also have sampled descendants. And you can imagine that maybe it's just okay to assume that these aren't sampled ancestors, but we can also consider situations if we don't allow those samples to be sampled ancestors, what we end up doing is inducing a large amount of speciation events and sampling, changing the distribution of sampling in order to accommodate the fact that that might be true. So here, if you look back to our reconstructed tree here, if we force those to not be sampled ancestor, what we see is a lot of more speciation events. And what that will then do is lead to induced higher a bias in the rates estimated for speciation, extinction, and sampling when we estimate under these models. So that becomes quite critical in accounting for that. Importantly, that we can contrive of certain data sets where this might actually be true. And that's because a lot of these birth-death processes are also applied completely, in the same way as they are to the fossil record, to data sets of rapidly evolving infectious diseases. And so when we've looked at these similar models in data sets from HIV, we see very, very, very low probability for sampled ancestors. And that's because when a sample is taken in HIV, a patient either changes their behavior, goes on antiretroviral drugs, or has actually passed away. And so the probability of transmitting to another host that might then be sampled again becomes extremely low in data sets where there's another behavior of the sample happening. So you can see that in order to do that, we need some external force structuring how these samples are taken. And obviously, we don't have that in the fossil record. A fossil doesn't get preserved, and then that doesn't mean that that's the end of that lineage forever, or it's no longer sampled again. So accounting for these sampled ancestors becomes really important. And so some simulation work that I've done with my postdoc April Wright is we've kind of looked at what is the probability, given that we have a model that's generating bifurcating lineages and we're sampling that tree back in time, what is the probability that we would get sampled ancestors, or what is the proportion of sampled ancestors that we would get? And so we simulated fossil data sets under 3 different sort of levels of turnover. So turnover is the rate of extinction divided by speciation. So you can imagine here in the case that we have relatively high turnover, so turnover is close to 1, so that means that speciation and extinction are fairly high. And here turnover is closer to 0, and so that's a situation where you actually have relatively low extinction. So in the case where we have relatively low extinction, then we actually have a very high probability of sampling ancestor-descendant pairs or assemblages. And that's because if you have very few of those lineages going extinct, they're obviously going to contribute lineages that can then be sampled later on. And this does change as we increase the rate of sampling back in time. And you can see that even as we get to relatively high turnover, and we could obviously do this for turnover where extinction is higher than speciation, but this becomes problematic in simulation. But and that's why we have extinction always being slightly less. But in this case, what you see is that even with relatively high extinction, we see a really significant proportion of those samples being sampled ancestors. So this really demonstrates to us that accounting for this is going to be fairly critical, because ignoring it is a clear violation in the model. And so what we did was just looked at a set of these simulations and evaluated the estimates of node ages in those data sets. And so here's just a single tree where we compared the different estimates of their node ages when a model that allowed for sampled ancestors and a model that did not allow for sampled ancestors. And the black line here in every case is the true node age. And you can see that for all of these, if we don't allow for sampled ancestors, that we're biasing our node age estimates to be far too old. And that's because either we have to explain the lack of sampled ancestors by making the tree very old and also forcing the sampling rate to be very low, or if we do, then we get a better estimate of those node ages. And as I mentioned before, if we in the same way, if we force there to be no sampled ancestors, then we're also going to bias our estimates of diversification parameters. So these include things like the rate of diversification, the rate of turnover, and the sampling proportion. But especially the turnover is highly inflated when we don't account for sampled ancestors. So here in the situation where we have fairly moderate turnover, we get a very high rate of turnover estimated when we don't allow for sampled ancestors. So this becomes an important part of our model that, when accounting for this fossil sampling back in time, becomes critical to integrate into that. So this brings us to the general interest of applying these, and we're kind of working on the development of these various tools for doing this fully integrative Bayesian analysis. And with this tree model, we can co-estimate divergence times and relationships with this cohesive application of a Bayesian framework. So to apply these, we recently looked at these approaches and applied them to a data set of penguins. And so penguins are very charming, but they also have this a really nice fossil record. And so this gives us a really good model clade in which to address some of these issues. And what we were interested in is how does our understanding of the diversification of penguins and the timing of the evolution of penguins change when we consider both the extant and the fossil taxa. So if we think about penguins and what we might be interested in their diversification, we can look at previous studies that estimate their divergence times using sort of standard node calibration approaches. And this gives us some picture of this diversification through time. But it's very limited to the crown group of penguins and those penguins still living today. But if we look at the history of penguins within the broader bird phylogeny, the closest sister taxon to penguins are birds like terns and albatross, so the tube-nosed birds. And recent studies that were estimating divergence times across the bird tree estimated that the most recent common ancestor of penguins and their sister lineage goes back into the late Cretaceous. So if we're interested in the diversification of penguins or the evolution of the penguin body form over time, ignoring this part of its evolutionary history becomes a critical oversight in terms of getting at these types of understanding the diversification of this group. So we see in living penguins a lot of distinctive morphological characters that give us information about what makes a penguin, how they associate with their environment and life history. And so this includes things like in or bone morphology, we see these flattened paddle-like scapula that indicate that because penguins actually fly essentially through the water, they have to actually move a lot more of this, use a lot more force to move the substrate through, in which they're traveling through, as opposed to flying birds that have these very thin blade-like scapula. So we see this, in living penguins as well as in some fossil penguins. And one of the things we're interested in is, what are the evolutionary processes driving, the evolution of these types of characters. Additionally, we also see that penguins are actually not very good at walking on land, and that's, one might be attributable to this very short and square-shaped tarsometatarsus, which is this fusion of the ankle bones that birds have. And so flying birds have these really elongated ones, and all of these penguins have these really short, blocky ones. And so this gives us an indication of, given that we might find some of this morphology in the fossil record, it allows us to sort of recognize things as penguins. Additionally, penguins also have these like flattened and solid wing bones, which conveniently, if you do study birds, they make for nice fossil records, unlike a lot of other birds. And so and they're living in nearshore marine environments. So with all of these really distinct morphologies, we see these present in the fossil record, but we don't see them in these other volant birds. And so this is important, I think, to understanding how this group has diversified. And so if we look at the crown group of penguins, we see a few fossils that fall within the crown. And these all bear a lot of the same characters as the living penguins that we see today. And so, even if we most of the previous studies that use these node calibration approaches just use the fossils that fall within the crown because they're limited to only using these fossils good for node calibration. Therefore, if we don't account for the rest of the history of this group, then we also still, even if we might be using these fossils, we still aren't really understanding what's going on. And that's because we see penguin fossils back quite farther than we've estimated the most recent common ancestor of living penguins. And so that includes this Kairuku penguin, which was about 1.5 meters tall. And it actually has some aspects that are similar to the living penguins, but has a scapula and a tailbone that are different from the crown group of penguins. And this fossil was dated to about 27 million years ago. And you can see that it's actually really nicely complete for a fossil that old. We also see, and so this is the oldest fossil penguin that has been described. And so this one was from about 60 million years old and years ago. And what's interesting here is that this penguin actually has more intermediate wing morphology that's somewhere in between flying birds and penguins. And so this gives us a really interesting picture about the evolution of this group and how they evolved in relation to their flying relatives. So if we look at the full diversity of the penguin fossil record, there's actually an immense amount of data and information that we can account for that we haven't been able to encompass in phylogenetic approaches before, especially phylogenetic approaches in a time scale. And so within penguins, there are 19 living species of penguins, but there are over 50 described fossil species. And also several more that haven't been described. And so this is something we were interested in, and given that we have these methods that can now unite this entire data set, how will that give us a better picture of penguin evolution through time? And so what we did is we use these fossilized birth-death approaches to co-estimate the tree of all our living and fossil penguins using 19 extant species and then 36 of the fossil species. We had about 200 discrete morphological characters and about 8,000 nucleotides. And so we applied these approaches particularly in the iteration of BEAST2. So this was work that was led by Sasha Gavrishkina, or Alexandra Gavrishkina. She's now a postdoc with Tanya Stadler. And so this is a tree that we estimated. And so one thing that it is important to communicate, especially when showing sort of summaries like this, is that summarizing these types of parameters or in a phylogenetic sense is very difficult at capturing the uncertainty that we have in this tree topology. And that's just because it's just very difficult to present visually. And so I do want you to get a sense that a lot of these relationships have very low statistical support, low posterior probabilities, and that's because we actually don't have a lot of characters contributing to the relationships of the stem fossil penguins. You know, we have a lot more stronger support for the relationships in the crown, and that's because we have data from molecular characters. But we do have this much better picture of our estimates, especially of node ages in this group. And so that's what was particularly interesting to us, and especially so Daniel Sepka, who is a collaborator, is a well-known penguin paleontologist, and given that we have this fossil record, it was always kind of a question to people who were familiar with the fossil record of penguins about the estimated time of the most recent common ancestor of penguins. And that's because previous studies, so here, have shown that the estimated age of the most recent common ancestor of living penguins to be in somewhere in about the early Miocene, and so, or even older. So these are estimates from previous studies, and so this study was the most recent analysis that used a Bayesian method and just node calibrations to estimate that age of the most recent common ancestor. And so in our analysis, when we then incorporated all of this information, so we have morphological characters contributing to our estimates of branch lengths. We have the fossil occurrence times contributing to informing our model on the diversification processes, as well as the relationships and the times. And then we have our various other models that sort of unite this all together. And given that we can account for all of that, we actually are getting a much younger estimate for the most recent common ancestor of living penguins than previous studies had. And this is actually more consistent with our paleontological understanding of the rock record and the fossil record associated with this group. And so given that we can then account for this history, we get maybe a more comprehensive understanding of the diversification of penguins, even of living penguins. And so it sort of did occur to us that what is really contributing and driving this younger estimate of this most recent common ancestor of living penguins. And so one of the things we looked at is when we just eliminate all of the stem fossils, what is the estimate of that age when we don't account for this diversification happening along the stem? And so when we just do the same analysis but without these taxa, we actually do also get a much younger or older estimate of the most recent common ancestor of penguins than we did when we account for all of the stem diversity. And so this indicates to us that there's strong information content coming from the fossil ages and other information, other data from the fossil record that's contributing to these inferences. So with a lot of this work, we're really interested in how can we improve our mechanistic model of fossil sampling back in time and diversification in general. Especially when considering the fossil record, it's obviously quite critical to account for variation in sampling of the fossil record, and particularly with regards to stratigraphy and how the structure of the fossil record really implies a lot of restrictions with regard to sampling. And most of our approaches assume that we have this very constant sampling back in time, but we know that's not true for fossils. And so what a lot of us are working on now are trying to extend these types of models to account for the stratigraphic sampling of fossils, but also biogeographic sampling of fossils. And so that's work that I have ongoing with a number of different collaborators. And there's a lot of things when you start working with paleontologists that you realize that either are really interesting data and from a perspective that we don't normally take within phylogenetics and systematics, that really requires some rethinking in how we're applying these types of approaches. And so we're also working on similar phylogenetic approaches to accounting for more paleostratigraphic range data that we have in the fossil record. And so this really kind of gets at this overall general motivation of a lot of us, and that's to get at developing statistical approaches for much more comprehensive and integrative phylogenetic inference, where then we can, given that we have data from various sources, and all of these data are conditional on the evolutionary relationships of taxa, and we can link these data together in a single integrative model, and then co-estimate these phylogenetic parameters in order to account for our uncertainty in all of these different approaches. And most of us have done these, especially macroevolutionary analyses, that rely on some either previously estimated phylogenetics tree and divergence times or distribution of those of that parameter in a more serial sort of framework. And what we're trying to do is to integrate that and to not account for the to account for the non-independence between these characters and these data sets. And so this kind of is the idea of this like statistical holy grail that a lot of us who are developing Bayesian methods have, is that at some, given that we have some set of data and some good generating models for describing how we might obtain those data or how those data might, have come into existence, then we can co-estimate all of these types of things to get at questions like body size evolution over time and how that might correlate with things like paleoclimate or water levels or biogeography, and have a single holistic analysis to account for this. Obviously, these are really challenging problems that might not necessarily be easy to do today, but we do have components of all of this, and we're now trying to work on ways of really understanding what are the limitations of our methods, or where can we make assumptions that we know are clear violations of our models, and really getting at how do you best approach these things in a practical sense. And so that's kind of a lot of the underlying motivation and direction that a lot of my work is going, and kind of where it probably will go for hopefully a while. And so with that, I'd like to thank you all for your attention, and just acknowledge my various collaborators and co-authors and funding sources. So thank you so much. So I'm happy to take questions. Sure. Sorry.
I really applaud your extremely ambitious and idealistic approach. And my practical concern is that for most groups of organisms, we don't have such a nice record as a penguin. So then if you have all the nice model here, how are you going to really applicate on the real ground?
So the fossilized birth-death process will also kind of, if you had two fossils, it would still make sense to use that. What you'll get in the end is essentially, a level of uncertainty, so credible intervals that are reflective of the fact that you have no data. Whereas previous approaches using these node calibrations, your uncertainty is entirely based on these priors that you construct. So what is interesting, in terms of the application of this in two clades where you don't have a lot of data, is that should be reflected in your estimates of uncertainty. So that would work, but it just, it becomes less satisfying and less practical, but it still would work. And ultimately there are some questions that without the fossil record or without adequate fossil record, you can't answer. And I think that even if we might be really interested in certain questions, if you don't have the data, you can't answer those questions. So, there are definitely cases where, these approaches are only useful for, groups with decent fossil records. But in some cases, like with plants and stuff, like, we have, some fossils and we have different types of fossils, and maybe we can account for how those might be distributed in our data set. And so the key also is in incorporating this idea of sampling and how the sampling of the fossil record changes across the tree, like so where we have groups of organisms that have high preservation potential and groups that obviously do not. And so given that we have those situations, we have to account for those too. I mean, definitely it's not as satisfying for groups that aren't penguins, but it's definitely part of the challenge of addressing data sets like that. Yeah.
As